In this lecture, we're going to spend some time talking about concepts that are going to be central to the rest of the semester. And it's all about how T cells interact with antigen and how T cells recognize antigen, and then what happens, well, not really what happens after that, but how antigen is prepared for T cells, and then potentially how B cells and T cells interact and how T cells interact with other cells. So it's a very, very central set of concepts. And it starts out with antigen processing and presentation. Up to this point, we've really been talking about whole antigen, or antigen as a whole. We did look at discontinuous versus continuous protein antigen a few lectures ago, but largely we've been just talking about a whole bacteria cell being recognized or being coded with an opsonin or such. But T cell receptors are not only specific in the antigen that they recognize, but very specific in the way that they recognize antigen. And T cell receptors can only recognize tiny little peptides, short peptides, maybe 8 to 25 amino acids long, of a pathogen. They're not going to recognize a whole piece of a cell. They're not going to recognize any carbohydrates. They're only going to recognize peptides. And these peptides are not going to be folded in their native protein state. They have to be processed. And so they go through something called antigen processing. It is a degradation of proteins by certain cells. And then these processed proteins are moved to the surface of a cell and presented to T cells. And then T cells, through their T cell receptor, will recognize that processed protein, and that's antigen recognition. And so antigen recognition not only requires interaction between recognition of the T cell recognizing the antigen, the T cell receptor, but also a human protein that's presenting it. And we call this antigen presentation. This is where a different cell will take this process that has processed the antigen and made it into tiny little peptide pieces, fragments, added it to or loaded it onto this human protein, puts it out on the surface and displays it. That's presentation. There are a few cells that are really, really awesome at antigen processing and presentation. And those are macrophages and dendritic cells. B cells are also going to be included, but at a later point. So when bacteria cells get in, or maybe a virus particle is taken up, they are going to be brought in by macrophages and these myeloid dendritic cells, processed, chopped up, degraded into protein segments, loaded onto this protein and moved to the surface. So let's go ahead and take a look at that in a picture form. Okay, so we have this pathogen gets into a cell. It's either taken up by phagocytosis, or it is a virus that has infected a cell, a dendritic cell, say. It's chopped up into pieces through antigen processing, and it's loaded on this human protein, and this protein is called an MHC molecule, major histocompatibility complex, MHC. We don't ever use the whole term, we just say MHC. This is a human protein that everybody has. Everybody has a set of MHC molecules. They're going to be very prevalent on these presenting cells, macrophages, dendritic cells, and they will present the peptide on the surface of the presenting cell. When a peptide is in conjunction with MHC molecule, only then can that peptide be recognized by a T cell receptor. And so you can see in this last image, we have our T cell over here with our T cell receptor. In this case, it's an alpha beta T cell. It's recognizing peptide antigen in conjunction with MHC molecules. Now, cells that have MHC molecules and do this presenting are called antigen presenting cells, or APCs. Okay, we'll come back to that concept a little bit later. Let's go back to something that we talked about, I mean, maybe like on day 1 of class or day 2. And that is the 2 broad classes or 2 major ways that antigens can be broken, or pathogens can be broken up into. We can have intracellular pathogens and we can have extracellular pathogens. Remember when I said it's going to be very important to be able to tell the difference between an intracellular versus an extracellular pathogen? This is why. They're treated differently and they're recognized differently. And that means that they will drive a different type of response by the immune system. So intracellular pathogens, mostly viruses. We do have some bacteria that are intracellular, but mostly viruses. And there are some parasites that are intracellular as well. But this is definitely the biggest of the 3 intracellular pathogens. And recall that they are going to be inside the cytosol, not inside of a vesicle, not inside of a phagosome of any type, but they are going to be in the cytosol. Because of that, they are going to be degraded in the cytosol through proteostomes. Okay, so there's little proteostomes in here. They're little barrel type things that'll just break these guys up right in the cytosol. This is different than what happens with extracellular bacteria or extracellular pathogens. We're looking at a lot of bacteria, we're looking at a lot of fungi, and we're looking at almost all the parasites. Because they are extracellular pathogens, they are taken into a cell via phagocytosis, endocytosis, and they are going to be degraded inside of a vesicle, inside of a phagolysosome, probably. Okay, so 2 types of pathogens, intracellular, extracellular. There's going to be 2 major types of MHC molecules. MHC class 1 molecules, MHC class 2 molecules. Well, that's handy. If there's 2 types of MHC molecules and there's 2 classes of pathogens, right? You see where this is going. MHC1 molecules are going to be important in presenting antigen from intracellular pathogens. They are going to bind those peptides in the endoplasmic reticulum and deliver them to the surface of that antigen presenting cell. On the other hand, MHC class 2 molecules are going to present antigen from extracellular pathogens. Extracellular pathogens are degraded in an endosome, and so MHC class 2 molecules are going to bind those degraded products, those tiny peptides, in the endosomal vesicle and deliver them to the surface. Okay, so I don't ask you to commit a lot of things to memory. This is something you're going to want to commit to memory. Somehow, some way, associate intracellular pathogen with MHC class 1 and extracellular pathogen with MHC class 2. If you can somehow do that, it's going to help you out along the way, because we're going to be looking at this time and time again, and this is working knowledge that you need to have in order to fully understand how the immune system eventually will respond to all of this. So MHC class 1, intracellular. MHC class 2, extracellular. we saw that T cell receptors have very, very specific binding to antigen. We saw that immunoglobulin has very, very specific binding to antigen. MHC class 1, MHC class 2 molecules have what we call promiscuous binding. They don't have strict specificity for the tiniest little bit, but rather they will bind intracellular pathogen if it's MHC class 1, extracellular pathogens if it's MHC class 2, but really it's just going to grab some protein from those different spaces and present it. The protein fits, it sits, kind of like a cat in a box, right? It's not highly specific. So it's promiscuous in its binding. And that's great because we only have a few MHC molecules to pick from across our repertoire of cells. Okay, we have two types of pathogen. We have intracellular, extracellular. We have two types of MHC, MHC class 1, MHC class 2. We're going to divide our T cells into the first division. And that is going to T cells. Okay, that's our first major division of T cells. And so if we have two types of pathogen, peptides presented in conjunction with MHC class 1. This would make sense because cytotoxic T cells will pretty much kill virally infected cells. Viruses are intracellular pathogens. They're going to be presenting their peptides on the surface in conjunction with MHC class 1. And so cytotoxic T cells will interact with MHC class 1 through their co-receptor CD8. So here's another thing to commit to memory. Cytotoxic T cells are also called CD8 positive T cells. CD8 is the co-receptor that recognizes MHC class 1. Okay, let's set that over there. Let's pick up the other set of T cells, helper T cells. Helper T cells help the immune system by really conducting everything. They are going to recognize antigen presented by MHC class 2 molecules. They are important in defending against extracellular infections. They express a co-receptor called CD4. CD4 recognizes MHC class 2. Now, the functions that helper T cells have are wide-ranging because there's a lot of different types of helper T cells that we'll look at eventually. But they are going to, help macrophages be better at phagocytosis. They are going to secrete a lot of cytokines to tell a lot of other cells what to do. And these are the T cells that are going to help B cells make better antibody through a couple different processes, but one of them is going to be isotype switching. So interact with MHC. Here, I can go to this slide. CD8 binds MHC class 1. CD4 binds MHC class 2. These are called co-receptors, and they go along with our T cell receptors. So this is a fantastic image for you to think together. Extracellular, or let's go intracellular pathogen, is taken up, processed, and presented on MHC class 1. MHC class 1 interacts with CD8 positive T cells and T cell receptors intracellular pathogen, CD8 T cells, MHC class 1, those three things together. Now, we very seldom call them, okay, we will sometimes call them cytotoxic. Sometimes we'll call them cytotoxic T cells, but that's the long name. So we can also call them CTLs for cytotoxic lymphocyte, or we can call them CD8 positive T cells. So those three names are used interchangeably, and depending on what you're reading, who you're talking to, what the context is, any one of those three names can be utilized to describe a CD8 positive T cell or a cytotoxic lymphocyte. Okay, but associate CD8 positive or associate CD8 with intracellular antigen, and MHC class 1. Those three things go together. Now, for the other side, our CD4 positive T cells. Sometimes these are called helper T cells. You're going to find, though, that calling them helper T cells eventually isn't going to be very useful. So for right now, we can call it a helper T cell, or sometimes we'll call it a T helper cell. But at this point, and eventually, calling it a CD4 positive T cell is probably a better action, probably a better thing to do, because there's going to be a lot of different types of CD4 positive T cells that we get to. But anyway, so those can be utilized to call. They're all the same, depending on the context and who you're talking to. But if you can associate CD4 with extracellular antigen and MHC class 2, those are the things that you're going to want to associate together. And again, really try to memorize that. Really try to commit that to memory, because it's just going to make things a lot easier as we get into how the immune system progresses. As a working knowledge, a really base knowledge that anybody coming out of immunology should have. Okay, so let's look at how this works in action again. We're going to spend a lot of time in the upcoming chapters talking about the immune response and what happens, but this is just reinforcing that a CD8 positive T cell will recognize the intracellular pathogen in conjunction with MHC class 1. So the CD8 is recognizing MHC class 1, the T cell receptor is recognizing the processed antigen, and it is highly specific and is going to be recognized on any cell that is expressing the MHC class 1 with antigen. And so then that will lead to direct killing of that cell, because it's going to be virally infected. The cell has to die. Now with CD4 positive T cells, there's really, so this is really any cell of the body. Any cell of the body mostly is going to have MHC class 1 on their surface, because essentially any cell of the body can be infected by a virus. But CD4 is not only, or not CD4, MHC class 2 is only going to be on a few subset of cells, and we call these antigen presenting cells, or professional antigen presenting cells, or APCs. Antigen presenting cells. Macrophages, dendritic cells are going to be the big ones. And then what happens is that a macrophage will take up an extracellular bacteria, process it, present it, load it onto an MHC class 2. The CD4 co-receptor will recognize the MHC class 2. The T cell receptor will recognize the processed antigen. That will get the macrophage to interact with the T cell. The T cell will then do its helper job and secrete cytokines to activate the macrophage. Eventually, we'll see how then that T cell can interact with the B cell, which also has MHC class 2 presenting antigen. And then the helper T cell can secrete cytokines to tell the B cell, hey, you need to up your antibody game. Let's get some new isotypes out there. Let's get some better specificities, and let's kick this bacteria out of the body. But again, we'll get into that in depth. Let's go back to our MHC molecules. The structure, I don't care so much that you know the structure of MHC 1 and 2 molecules. They are glycoproteins. We're going to see MHC class 1 on a lot of different cells. It's made up of 3 separate chains as well as beta-2 macroglobulin. MHC class 2 is going to be made up of 2 chains, alpha beta chains that are joined together. They're both MHC class 1 and 2 are going to have membrane spanning domains, but essentially the important things to remember here is that MHC class 1 is going to bind intracellular pathogen, process pathogen, where MHC class 2 is going to bind process extracellular pathogen, and then they're going to interact with the co-receptors on a T cell receptor. Now, notice we're not seeing our CD3 complex or our zeta chain. It's there. It just makes the diagram too funky. But just know that there are other proteins in association there. Okay, so CD8 is going to recognize the alpha chain on MHC class 1, and CD4 is going to recognize the beta chain on MHC class 2. I don't care, again, that you know that. The big important part is that you connect CD8 with MHC class 1 and CD4 with MHC class 2. We call these cells down here differently, a target cell, because this is going to be infected and it's going to be a target for cell death because cytotoxic lymphocytes are going to kill virally infected cells. We call this an antigen presenting cell because it is not infected, but it is the cell that uptook the extracellular pathogen, processed it, and is now presenting it. And so that's why it's an antigen presenting cell. Okay, here's a kind of a summary slide showing you MHC class 1 and MHC class 2 processing differences. I talked about the proteostome inside of the cytoplasm here. It's just hanging out. It's a protein that will take up any intracellular pathogen viruses, chop them up into these tiny little peptides, move them into the ER, and then that is where the MHC class 1 is going to pick up, load that peptide onto its little loading zone, and then it will be moved to the surface of the target cell, presenting this processed peptide from an intracellular pathogen saying, hey, I'm infected. Look at this protein I have in me. CD8 positive T cell, come find me and kill me. MHC class 2, phagocytosis, is how that extracellular pathogen gets brought in. We talked about the processes that occurred during phagocytosis and the degradation with the phagosome binding with the lysosome, creating tiny little peptides that then get loaded onto an MHC2 molecule right inside that endosome. And then it's going to be moved, once it's loaded onto the MHC class 2, to the surface of the cell. And this is an antigen presenting cell because it's not infected, but rather it took up the extracellular pathogen via phagocytosis. Then we will have a CD8 positive T cell recognized, and we'll have a CD4 positive T cell recognized. That antigen piece. Okay, so here, the human cell consists of 2 components or 2 compartments, right? This is review. We talked about this. We have our nucleus and our cytoplasm. And then we have all this extracellular space that, is created by all these vesicles. Okay, so the nucleus and the cytoplasm, that's kind of all considered to be part of the cell. It is like the body of the cell, where this vesicular system is made up of extracellular components that get brought in through endosomes and then will be shunted out through like some sort of endosomal vesicle get moved out. And then we also make proteins inside of the cell. So we have transcription and translation happening on the ribosomes, or either on the endoplasmic reticulum or out in the cytoplasm. Those then get put together into the Golgi and then moved out, and then proteins are excreted out, right? So that's just cell biology, nothing specific to immunology here. But when we talk about how this then relates to immunology, we have proteins, whether they're of pathogen origin or self-origin, or we can say self-origin or non-self-origin, they can still be degraded inside of the cytoplasm. And because the cell doesn't have like the brain to differentiate a non-self peptide versus a self peptide, both types of peptides can be loaded onto an MHC1 molecule. It's promiscuous binding, and so we can have self-peptide pieces, we can have non-self-peptide pieces being displayed at any given time on a cell with MHC class 1. The difference, though, between a healthy cell and a virally infected cell is that a healthy cell really should only be displaying self-peptide. When a cell is infected, it's going to be expressing or displaying both self and non-self mind. And so a major function of the vesicular system of a cell is to take things up by phagocytosis, endocytosis, pinocytosis. It degrades them and then can utilize these nutrients, or it can also process them and bind them, if they're non-self, with MHC class 2 molecules. So then this peptide in conjunction with MHC class 2 molecules will be exported to the surface. If there's no extracellular infection occurring, then cells are only going to be they were brought in, but they're still self. But if there's an infection going, not only will self peptides be brought in, but also non-self. And then so again, you'll have that combination of self and non-self peptides being displayed by. Okay, we're going to stop there, and we're going to pick up with one more lecture to wrap up this topic.